Yeah, until recently, I was there almost full time and decided to retire in March and to pursue some commercial interests. But I'm very much in love with this organization. I helped build. I was there for 7 years, and we built this research institute that is probably one of the great stem cell institutes. In the audience, we talk about CERM, California, and we talk about the Catapult Group in Great Britain. We talk about CCRM in Ontario. We're a 501c nonprofit, so if you're looking for investments, the only investments you're going to get here is getting technology and getting things done right, and that's the organization that is New York Stem Cell Foundation. Let's see. Oops, how do I? Oh, here it is. Our mission is to accelerate cures, and we were set up 10, 12 years ago to fight the issues with use of stem cells. We're stem cell agnostic. We work, because we're privately funded, we work with all types of stem cells, including the embryonic and extremely rare cell types. We actually are one of the few people who do somatic cell nuclear transfer. We also have a huge investment in the area of investing in young people, and so we award funding in 5-year grants to young investigators to reach out. And I guess one of them would be, we talked about gene editing. Zhang is one of our early identifications. People when they're working in the CNS area, like Ed Boyden. So we have invested as an organization in very, very young people and recognize their work. We also have this outreach in symposia. So not unlike this meeting here, we have a very large meeting that's held in New York at the end of October, and I think I have a slide on that. But we also built the Research Institute. So for many of you in the audience who recognize me, I started here 30 years ago, that is in San Diego, and built some of the first 2 major gene therapy companies. And about 7 years ago, somebody enticed me and Susan Solomon to come to New York. And I thoroughly have said that I have lived both lives in 2 coasts, a beautiful place like La Jolla and an unusual place like New York. But the idea of building a research institute that was very disease-focused, using a platform technology of stem cells. And maybe I'll tell you what we've done with that. So we've created this community of scientists, and so it's really a research institute without walls. Part of our mandate was that if we invest in you as a young investigator, we actually have agreed with the institutions to get rights, both for use within our own institute, as well as some, if they are able to commercialize rights. So that has become very, very nice way for the organization to sustain itself. We have a commitment to scientists. It's alumni from all over the world. We have a reach through, and again, even though I think Susan has said this quite eloquently, even though we're called the New York Stem Cell Foundation, we're really international. And so her husband, who was a Pulitzer Prize winner, said, New York Times is a newspaper that's international in so many ways that for reasons that we had a name, and that's called the New York Stem Cell Foundation. But we are very global. But the thing I want to talk to you most about is about what we're doing here. So this is more, I'm not selling anything, I'm not telling you to invest in anything. I think of the Research Institute that we built as a resource for all of you to utilize as you advance the technologies, be it gene editing, gene therapy. The one thing we have is we have tools and reagents that are freely available to both the academic world and then, of course, with the commercial world, it's a negotiated agreement. We have this neural focus because in many ways the initial stem cell biology, the default pathways that we understood with embryonic cells was in the neurons. So we have built this large organization built on essentially CNS diseases. And those have become tools and very available to individuals. So we have collections in multiple sclerosis, different types. We distinguish this, and I'll talk more about this. We have a very large, we worked with Michael J. Fox to collect a large Parkinson's collection, which is linked to clinical work as well as Alzheimer's. We have a large grant to do this. So the vision was, could we take humans off the streets of New York who had genetic profiles and eventually sequencing and make personalized stem cells? And the answer is yes. And so what we've done is we built, but that represented a technological hurdle. That idea of every person being born or walking around could get a personalized stem cells was an issue of economics and scale. And so one of the first things we approached it, and it was very much from my manufacturing kind of thought process, is we derived an automated platform. It's a Nature publication, so I don't need to go into. You can always Google this. But we've been able to take this platform and we can automate the production of IPS, induced pluripotent stem cells, from any individual. So from that platform, we have now built a whole collection of cell types. Cell backgrounds, genetic backgrounds. So if you're interested in a Parkinson's panel, a rare disease panel, we have a very large ethnicity panel. We were able to do this. We expanded that platform from the automation of making stem cells into making differentiated cells. And differentiated cells are really what people are interested in. So many times when you acquire a cell line, it's usually through a third party. That third party says it's a blank cell line, it's in a catalog, some repository. As you know, it's been published recently in several journals. A lot of these lines are incorrect, especially the cancer lines. And so that has become an issue of curation. The nice thing about this is that we have been able to build this all based on new technology of barcoding, et cetera, that allows you to track the beginning, the birth of a cell, the acquisition of that tissue and or cell to the final product and freezing. So that's part of the automation. So this allows a lot of technological quality assurance and quality control, which is not usually available. And again, that's available to those in both the academic and commercial world. And I think we have currently 5 or 6 of the largest farmers in the world collaborate with us on various aspects of this. So consequently, since we had differentiated cells, we do disease and modeling. We also do this area of tissue engineering, and we also now bank cells. So the technology that we've had in the induced pluripotent cells has also been applied to the high throughput generation of embryonic stem cell lines. Turns out that we're the only group left in the United States that can take discarded embryos from in vitro fertilization clinics that people are discarding. So we've collected those as well to generate eventually embryonic stem cell lines for those who are interested. We've been highlighted in both many publications and research, so our science is intact. Think of us as a privately funded organization whose sole purpose is to enable researchers, and when we say researchers, and to help patients. And so when I say researchers, I don't care if you're an academic or a commercial. It makes no differentiation to this organization. A lot of collaborations all around the world and in the United States, so you can see some of these. But the differentiation, the technology, the fact we could actually work on things that was unfundable by the NIH. One of our advisors, a former head of NIND, Zach Hall, we tried to get one of these grants that allowed it. I think it was during the Obama administration, one of these things were, and we were laughed out of the room, but we did it anyways because it was a fundamentally good idea. It cost us $25 million, but thankfully donors saw the vision and when the automation was developed. We also have now, and will probably be in a new publication, where we can actually genome edit at scale, where we can take a lot of different lines and different targets and go automate. And again, it's through the robotic systems we've developed. So we've industrialized stem cell research, and this is the publication that was in Nature. So now we're really interested in connecting what I call the genotype to phenotype. And so it's in vitro GWAS studies. So we now have focused on fully sequenced people, for example, with George Church. We have essentially taken all the fully sequenced people that he has through the Personalized Genome Project and made iPS cell lines. So this is going to be very, very important for people interested in things. Again, with certain disease foundations, we've made, like the Batten's Disease Foundation, we've made several different panels of lines for people in those areas studying these things. So we have a platform. The key in our platform is probably our informed consent. I think our informed consent was one of the pioneering informed consents that allows us to gather both the genetic as well as the clinical information from patients, and now has been put into a repository where we have several thousand cell lines. The key is that the cells are available, the patients are recallable, so if we don't have full genetic information on them, we're quite capable of calling them back, and that's one of the keys to this. And given the fact we are centrally located in New York City, as I was told, there's no greater diverse area of the country than New York City. And I laugh at this because we had a problem we needed a Native American, so we were searching and we couldn't do it. We put up an ad in one of the school newspapers here in New York, and there are several colleges, and we got hundreds of people who claimed they were Native Americans, and then we were able to genotype them and actually get a sample for somebody who was interested in doing that. We've built a new research institute, and we moved in January, and I think my song was, I went to New York to do something. I think I finished it, but it's a new beginning. That new beginning is this wonderful group that has been, you see automation there and computers. It's on the it's on the west side in Midtown, near the Hudson Yards project. It turns out this building is going to become one of the new biotech buildings in New York. There's a huge commitment by the state. So those who are interested in locating a company in New York or laboratory facilities, we're the anchor tenant in this scenario. I wanted to highlight two really important things that we, just a recent publication we did, where people a lot of times had a tough time making microglia cells. I think our technology, the fact we can do iterative technologies that experimentally is difficult for a single postdoc or a set of technicians to do, the fact we can iteratively derive things. We're able to do two really important cell types, microglia cells, which may be important in the immune surveillance in the human brain for things as Alzheimer's, and an earlier publication in oligodendrocytes. The interesting thing about these, and there are other publications within the microglia area, we've done these for now 20, 30 lines. As a matter of fact, investigators from around the world, specifically in these lines, call us about these protocols because the robustness of what we have done, not only with the using, for example, in the microglia, we've done it for embryonic stem cells as well as induced pluripotent stem cells. The conference we have is at Rockefeller University. It's October 24th, 25th. It's a wonderful time to visit New York. I wish I could get you Hamilton tickets, which is really difficult. We were able to get one set from one individual last year, but I've got a lot of connections. My sister-in-law is on the theater board, so might be able to get you one, a set for a lot of money, unfortunately. And that's New York Stem Cell Foundation. Think of us as a resource to help you. It's not, we're a funding agency for young investigators, but more importantly is that we help you, that the researcher, get materials to do research on, which is one of the rate limiting steps for many of you in the field of stem cell as well as gene therapy. So thank you.